Friends, today I am starting the lecture series on the nervous system. I will be giving today about the introduction and general plan of nervous system. In the next slide, I have given the plan of presentation. It is as below. Number 1, description of the parts of the nervous system, the neuron. Today I will be dealing with. Number 2, physiology of somatosensory system, general plan, and then processing of a neo and paleo sensations, then processing at different levels, thalamus as a separate lecture, and cortical processing. Thirdly, I will provide or I will give a detailed account on the organization of the cerebral cortex and general overview, various areas. Then I will discuss about somatosensory cortex and its function. Fourthly, I will be talking about a motor system, and this motor system I cover the voluntary action, the motor cortex, corticospinal tract, basal ganglia, cerebellum, and control of axial muscles separately in series. Fifthly, I will be talking on postural reflexes operating at various levels of neural axis, because without the postural reflexes, the voluntary action is not possible. And spatial reference will be given to vestibular system and the writing reflexes and the hindbrain reflexes, that is the in decerebrate animals. Sixthly, I will be talking about the organization of the spinal cord and the spinal reflexes. Seventhly, I will be talking on hypothalamus, limbic system, and frontal lobe, and all the functions of the prefrontal lobe. In the next series, I will be talking about the sleep wakefulness, reticular system, and brain electrical activity, that is EEG. I will be covering memory and learning in the next lecture series, and then speech, various disorders of speech, and finally I will deal with the cerebrospinal fluid and blood-brain barrier. Some of the chapters which are not covered in the series mentioned previously, I will be dealing them separately in a separate series of lectures, that is about a neuron, classification, nerve fiber classification, physiological changes after nerve injury in a detailed manner, resting potential, action potential, genesis of action potential. Then I will deal with the synapse, the properties of the synapse, different type of synapses. Then I will talk about the spinal cord, organization, the spinal injury, spinal shock. I will give next series the autonomic nervous system, and finally I will cover all the spatial senses as far as possible. Hope with these arrangements you will have adequate information about the nervous system required for the MBBS students. I am describing different parts of the central nervous system, starting from the tail to the cranium, that is caudal to cranial side. The spinal cord, medulla, pons, cerebellum, midbrain, diencephalon, cerebral hemisphere. All these things are considered as a central nervous system, and there is another term known as a peripheral nervous system, The peripheral nervous system is that part that covers the nerves supplying the various parts of the body which are coming out of the spinal cord or the brainstem. These are part of the peripheral nervous system. Now I am describing the different parts of the brain. Is known as encephalon. Brain is divided into three parts: forebrain, midbrain and hindbrain. Forebrain is known as prosencephalon, midbrain is a mesencephalon and hindbrain is the rhombencephalon. These are the parts. In subsequent slides, I will describe these parts and their components in a detailed manner. Forebrain consists of two parts named as telencephalon and diencephalon. Telencephalon, tele means end, encephalon is brain. This is the end part in terms of the evolution, end part in terms of receiving the inputs where it is processed. The telencephalon consists of the cerebral cortex, all two hemispheres, and the various areas present here, including the hippocampus, amygdala, and those parts of the limbic system. It also includes the basal ganglia and the olfactory bulb. The diencephalon is the part between the base of the brain and the midbrain, and this is governing number of important functions that connects the endocrine system, the pituitary. It is having two parts, thalamus and hypothalamus. Thus, diencephalon plays an important role in controlling the neuroaxis. Pineal gland, part of the diencephalon structure. The midbrain or mesencephalon comprises of three important structures: the colliculi, tectum and tegmentum. This is the structure caudal to the thalamus and rostral to the hindbrain, that is pons. And this structure I have shown on the right side in a detailed manner, where you have superior colliculus, inferior colliculus, tectum and tegmentum. And this is the part which connects the entire cerebral cortex to the pons and medulla. The hindbrain, rhombencephalon, this comprises of the pons, cerebellum and medulla. You can just see this is the component of the midbrain here. This is cerebellum here, and it is connected with the pontine and medullary regions with the cerebral peduncles. So now we have the pons here, and this is the medulla. And the naming cerebellum and pons together are called metencephalon, and medulla oblongata is called as myelencephalon. Why I am trying to emphasize on these terminologies, because whenever we are trying to say certain words, you may come across these words and you may have to recall these words at that time. So for that reason, I am trying to prepare you for understanding these terminologies. Let me repeat here again. We have forebrain, we have a prosencephalon, and then we have a telencephalon and diencephalon there. Telencephalon is the cerebral cortex, then midbrain, that is a mesencephalon, then rhombencephalon that comprises of a metencephalon and myelencephalon. Okay, with this maybe I may just summarize. The brainstem forms medulla, midbrain, excluding the cerebellum. Those are the components of the brainstem. There are other terminologies like the use of the various the planes in the nervous system. So maybe you must have been you must have studied these things in anatomy, but however, to recall and recapitulate all those things, I'm providing the recap. So in case of man, I will not mention about the rat there. You can just see the tail end and the rostral. Tail end, the rostral, that is rostral to caudal. Then from the top, this is the rat sitting here. On the top is the dorsal and the limbs where it is ventral. So if you come back here in human being, here it is in the cranium. You can just see that the frontal region above the nose, that is a rostral end, and the back of the skull is a caudal. That is how it is taken. And the superior and inferior, or you can say that on the top, that is the dorsal, and bottom is the ventral. So this is the another nomenclature. You can just see that this is the dorsal, and this is the ventral aspect. Similarly, so from the middle of the brain to the tail in the spinal cord, that is the rostral to caudal or cranial to caudal, whatever you can say. Now we just look into this here, the dorsal and ventral. In case of spinal cord, the dorsal is here on the back and the ventral is here is the front. You can just visualize all of that. You can just see number A is the horizontal section. When it is cut, say for example, if you make a section through the mouth in horizontal to the plane, so then you have the horizontal section, something like that. Or coronal section, that is cutting. You just see that cutting the head in the coronal, along the coronal sutures, that is a coronal section. You can just see, you can visualize this similar thing here in this thing. This midline, these are medial structures. On the lateral side, these are lateral structures, medial structure here and the lateral structures there. So that is the sagittal section is along the longitudinal axis of the brain, where the sagittal suture is there, and if you cut, you will see such a type of visibility. So you just try to study these things, try to understand these. Now let us go back. What are the various cells of the nervous system? The major cell of the nervous system is a neuron. It is functional unit of the nervous system, and that perform all the functions, whatever the actions we perform, that is entirely performed, processed and dictated by the neuron. To support neuron, there are a number of non-neuronal cells, what are called glia or neuroglia. They include astrocytes, oligodendrocytes or Schwann cells. They provide a myelin sheath. The oligodendrocytes cover the myelin sheath for the central neurons, and the Schwann cells provide myelin sheath for the peripheral nerves. So the Schwann cells are there in the peripheral nerve, oligodendrocytes in the central nerve fibers. So there are other group of cells. These are called radial cells. These are progenitor cells. And these progenitor cells are of great help. They are in a very small number, very small number, and they can be used for the neuronal stem cell, for harvesting the neuronal stem cells, the radial cells. Then we have the ependymal cells which cover the cavities, the layers. These are ependymal cells. We have microglia. Microglia are the cells which are reticuloendothelial, derived from the reticuloendothelial cells, and they perform all those reticuloendothelial functions and many others. If you look at the numbers, these non-neuronal cells, that is glial cells, are more than the neuronal cells. They are, because they perform a number of other functions for the nervous system. So all these cells, whether neurons, glia, are organized in a definite fashion to execute the action perfectly. So that is the precision at which these are organized that has to be wanted enough. Now coming back, the parts of the neuron. So you have three major components. One, dendrites. Two, the cell body, the soma. Three, the axons. The dendrites are the projections, the small projections coming from the cell body. They will be spreading all over, and these are exactly the antennae, or the receivers. They receive all type of inputs, and once they receive inputs, they provide the inputs to the soma or the cell body for processing. So now these dendrites, they have a number of ramifications. Suppose if I am performing a particular sensory modalities, the dendrites will arborize, dendrites will increase, and we have a primary, secondary, tertiary dendrites coming up. You can just see there, the number of branchings you have. The more branching is more evolved. That means that they are trying to sense each of the sensory modalities, or the whatever the modalities, to be processed precisely. Now they will have spines, dendritic spines. Again, these are little protuberances where axon of other neurons synapse. So these the dendritic knobs or dendritic spines, they will have a contact area receiving inputs from other axons coming to these neurons. Then comes the cell body. Cell body is a large structure. Depending upon the type of the cell, their shape, they will be different, but they basically have all the material necessary for synthesis of neurotransmitters. That is a protein synthesis. Then they have the nucleus, the nucleoli, mitochondria, endoplasmic reticulum, Golgi complex, and this RNA particles, that is the missile substance, that are called the chromatophilic substance. In addition, these cell bodies, you can see that they are having the microtubules, the transporters, the channels, tracks, in which these microfilaments, they transport the material synthesized in the cell body to axon. So that means they have the microfilaments, the vesicles, the mitochondria, and all other structures necessary for performing or the synthesizing, metabolizing, and packaging and transporting the neurotransmitter. Then the cell body then leads to a hillock or a more like a funnel like thing. It funnels down to a narrow. This funnel head is the what is called an axon hillock. From there the funnel tube comes up. This is the axon. Now this axon, the initial segment of this axon hillock, that is an important segment for the activity of the nervous system. This is the site where the activity begins, the action potential begins, because if you see number of synaptic contacts which are developed on the dendrites, on the soma, their activity all summed up, whatever the synaptic activity, as it is the total algebraic summation of all the activity coming from various dendrites and the soma, and that will be displayed in the, or that will be initiated in the initial segment where the action potential generates. That is where the soma communicates or transports or transmits the impulse to the terminal segment of the axon. So the axon, it contains the plasma inside, and it is covered by the membrane exolemma, and that exolemma is covered by the myelin sheath. You can just see the myelin sheath there, Schwann cell or oligodendrocytes. If you have one Schwann cell is surrounding the peripheral axon, whereas in case of an oligodendrocyte, these are central axons. They are covered. One Schwann cells just embraces a number of other neuron or the axons going up. So that means there will be multiple axons are embraced by the oligodendrocytes in the central fibers. So then we have the presynaptic terminal, and then in the presynaptic terminal, that is the bulge, there will be a button or a knob, and in this knob you have a number of synaptic vesicles and various vesicle associated proteins for the transport or the release of the neurotransmitter. Site where the action potential is generated is the axon hillock, and that is very close to the cell body. These are the various parts of the neuron. I have drawn the diagram, and you can also draw it from your own. Now I am providing broad idea about nerve injury, because we may come across certain terminologies. Seddon categorized the nerve injuries in three broad classes, and they are named as neuropraxia, axonotmesis, neurotomesis. Neuropraxia is a mild injury without any severity, that is, nothing is disorganized, nothing is cut. However, there is small changes in the myelin structure, and there is no loss of continuity, but there will be transient functional loss that may be due to some ion induced changes which may block the conduction, as happens in case of a Saturday night paralysis, wherein person sleep over his arm and there is a pressure on the brachial nerve or radial nerve, and he will have the paralysis, something like that. This is neuropraxia, no injury, it is mild form. Axonotmesis, it is due to the interruption of the nerve axon. Nerve axon is cut. However, surrounding myelin or surrounding perineurium or epineurium are preserved. So again, there can be 3 or more classes here, classes which can be categorized here. But however, the myelin is lost, or perineurium is lost, or epineurium is lost. But overall epineurium is preserved. So that means structural continuity is there. Inside core of the axon is damaged or severed. And you will see these things in crush injuries. Third one is neurotomesis. It's a severe degree wherein nerves are severed, nerves are separated from the site. There is a total functional loss, and recovery without surgical intervention is difficult. What happens? You have to bring the nerve fibers in close proximity, wherein the spurting or the growth of these axons may attract the opposite end and they may unite. That is this thing. Otherwise, there will be a scar formation led by the fibrosis or some other thing. Then it is absolutely difficult. So this may happen in case of a traumatic cut injuries. What happens when nerve is injured? One, there is an immediate function loss. This function loss may be the loss in the sensation or sensorium, or loss in the motor functions, or any other functions that nerve is performing. The recovery may take place depending upon the severity of the nerve injury, as mentioned earlier, or maybe it can be restored by surgical intervention. There is a possibility. We can assess the nerve injury, what is the degree of nerve injury, if we start seeing some loss of Nissl granules. That is known as chromatolysis. And this begins within 6 hours of injury and continue further for some days and weeks. This would produce all the Nissl granules, which are rough endoplasmic reticulum and ribosomes. There is autolysis, and there is a swelling of the cell body, and there is a marginalization of the nucleus, and there would be changes in the apoptosis or apoptotic parameters, and finally the nucleus may be extruded from the cell body. That means in that case we cannot reverse whatever the effects. The chromatolysis can result from the nerve injury grade 2 and 3, as mentioned in the previous, ischemia, toxicity, cell exhaustion, and virus infections. Distal to the injury, there is another type of degeneration. That means the distal segment or a terminal segment degenerates, and this terminal segment, all the neurofibrils, neurotransmitters, they are excluded, they undergo degeneration, and they will be replaced by the myelin sheath. Myelin sheath would try to making a droplet like structure, and finally the phagocytes come in and phagocytosis take place. Fibroblasts also enter. There may be fibrosis of the entire injured distal segment, and it is seen in 2 and 3 categories of nerve injury, that is axonotomesis and neurotomesis. Now we will see the types of neurons. The types of neurons which are classified according to the histological structure. You can see the unipolar cell, that is one side cell body, the remaining side is axon and dendrite, as happens in case of an invertebrate neuron. Bipolar cells, present either in the retina or in the auditory pathway, that is after the hair cells we have a bipolar cells, or elsewhere. Then we have a peculiar type of a neuron which looks like bipolar, but really it is unipolar. That means one axonal projection, in between we have the cell body, as happens in case of the dorsal root ganglia, where it holds the cell body of these pseudo unipolar cell. And that will be one side, it will go to the skin or a muscle, on the other side it will go to the spinal cord. In between you have the cell body. Or you have multipolar cells. You have three examples here. One, motor neuron, you see how there is arborizations of the dendrites receiving inputs. You can see the pyramidal cell of the hippocampus. You can see the number of dendritic arborizations, how they are picking up various inputs, various things, and again they are apical dendrite, cell body, and basal dendrites. You look at that Purkinje cell, where you see number of dendritic arborizations like a tree you have, and all those tree elements, the big banyan tree, something like that, these Purkinje cells, and they receive all inputs coming from the horizontal cells there, or which is coming from the horizontal, that's the granular cells that will cut across all these dendritic arborizations. These are a number of types of neurons depending upon the histology. They can also be classified according to the function. Afferent neuron, efferent neuron, interneuron. Afferent neuron which receives information, efferent neuron which gives away information to the next site, and the one which is sitting there trying to connect between the afferent neuron and the efferent neuron, or a different type of neuron, different set of neurons, that's an interneuron, that is a modulatory neuron. Or the neurons can be classified according to the transmitter, they are named after the transmitter released. This transmitter, if it is releasing acetylcholine, cholinergic. If it is releasing glutamate, it is glutamatergic. If it is releasing dopamine, it is dopaminergic. If it is releasing adrenaline, adrenergic. Serotonin, serotonergic. Peptides, substance P, or these things may be peptidergic or substance P-ergic, whatever you can call. Or GABA, GABAergic. Enkephalin, enkephalinergic. Histamine, histaminergic, so on and so forth. The neurons are also named according to the shape. The basket, stellate, star-like, staghorn, like a staghorn only, pyramidal, horizontal, granular, they are making the granules of the cells look like a granules, fusiform, magnocellular, the big, the parvocellular, small, midjet, that is the small cell, and coniocellular, dust-like. So these are some of the classifications of neurons which you may come across during our course. Now looking at the different types of neuroglia or glia. Here I have mentioned 6 of them. Ependymal cells covering the various ventricles. You can see oligodendrocyte, one of the oligodendrocytes making the myelination, producing the myelin sheath, one cell trying to have little hands and covering the entire the nerve fiber. That happens in case of central neurons or central axons. Satellite cells, these are those cells which try to cover help, they cover the neurons, and they try to provide a nutrition support and whatever the material required. These are satellite cells, these are units which provide these things. The astrocytes, these are but in abundance. They are the major ones. They are in abundance. They are performing a number of other functions. You can just see there. Microglia, they are distributed. Again, they are more in number. Schwann cells, these are the cells which form the myelination in the peripheral nerve fibers. Neuroglial cells or glial cells constitute more than the neuronal population or the neuron number. So approx 10:1, 10 of these cells to one neuron. Briefly, functions of the various glial cells. I am mentioning only few of them here because which are of important. Astrocytes. They are the support cells to the neurons. They hold them together. They try to provide this base or a structure. Not only that, they maintain the milieu around the neurons. That is a scavenging or trying to clear away the excess of potassium which is detrimental to the neuron. Excess of hydrogen ions which is again detrimental. Excess of neurotransmitters, say for example the glutamate, if it is in excess, that may produce excitotoxicity, and all other metabolites accumulated in the interstitium where neuron is present, they will try to clear. That is one of those, maintains the homeostasis, right homeostatic environment for the neuron. Thirdly, they are responsible for the neurotransmitter released at the synapse by the neurons, because some of the neurotransmitters are taken up by the glia, they are degraded, they are resynthesized, and or they supply the raw material to the neuron for resynthesis. So that means they are actively participating in the neurotransmitter metabolism. So that means they give the stock, they keep the stock of these things. So that is one of the important function of the astrocyte. They are also important for maintaining the blood-brain barrier, because on one side the astrocytes are making contact with the choroid plexus or the endothelial cells. On the other side, they hold the neuron, they keep away the neuron, they make tight junctions, and so that nothing leaks into the cerebrospinal fluid. Thus, they maintain an environment which is conducive to the survival of the neurons. So that means they maintain the blood-brain barrier. They provide the scaffolding structure for the entire brain and the spinal cord organization. How these particular tracts, fibers, or the afferent neurons, the efferent neurons, or whatever, they have to be put them together. In addition, they synthesize a number of trophic factors, what are called neurotrophins, brain-derived growth factor, glia-derived growth factor. So there are series of growth factors coming from the astrocytes. They are necessary for maintaining the survival of the neurons and the growth of these neurons, in the sense the arborizations, the dendritic arborizations, the synaptic activity, and the transmitter functions. For all these things, these trophic factors are necessary. Then they also do some scavenging, that is a phagocytosis. They clean away the any degraded substance coming there or any foreign particle, they will be eliminated. Ependymal cells. These are the cells which line the ventricles. We have the lateral ventricle, we have the fourth ventricle, and then we have the whatever the canal. These are lined by these ependymal cells, and they also have some store of those progenital cells, that is the stem cells. These cells make a tight junction with the astrocytes because they make the junctions with the endothelium tight so that nothing leaks into the cerebral cavities. And they have a cilia so that they circulate the cerebrospinal fluid. These are the functions of ependymal cells. As I have already mentioned about the oligodendrocytes and Schwann cells, they are for the myelination of either the central neurons or the peripheral neurons respectively. Microglia, they are part of the reticuloendothelial cells, perform all the reticuloendothelial functions. In addition, they also do the phagocytosis. They try to maintain the local milieu. I recommend following books for reading. 1. Principles of Neuroscience by Kendall and Associates. You can go for the newer editions if you have. 2. Guidance Textbook of Medical Physiology, International Edition. 3rd, Genong's Review of Medical Physiology, again International Edition. Here are the assignments of this lecture. I have given 2 types of questions. 1, giving a brief descriptive, and the other filling the blanks. Answer these questions and assess yourself. In the next class, I will deal with the physiology of somatosensory system. Thank you very much.